All right, let's keep moving on. We've got two more mini lectures left to do. This one actually has very strong relevance because we're going to talk about phospholipids and how they fit in the membranes. And you notice that phospholipids can include sugar-based lipids, glycolipids, that can affect blood types. It turns out that blood types are determined by glycophospholipids, and basically glycolipids for sure. So phospholipids have phosphate. And just remember that phosphate actually can form two bonds to alcohols. Glycerol can provide one alcohol group, and then the other two can bind long chains. And in fact, on the other side of the phosphate, you can add another alcohol. This is what choline is, and we've talked about that before. So this is why they have the structure that we've shown you before, where you have the two chains, and then you have the glycerol in the middle as the backbone, and you have the phosphate with another head group on top of it. So what can be on the head group? We've already mentioned choline, and here finally it is. We have choline sitting there looking at you and showing you what the net charge is. By the way, there's a slight error on this version. I think it's fixed in your version of the textbook. But notice that right here it says that net charge is going to be 0 because the charge of this is related to the charge of the phosphate. And so ethanolamine, they have that right. This is an NH3 plus because it has 4 bonds. And so therefore, plus this minus phosphate, net charge is 0. Choline also has a plus, they just didn't show it. But they've shown it on other versions of this and realized that the net charge of choline is going to be 0. It's an ammonium type nitrogen in the middle there. Serine, on the other hand, you can use the serine side chain as the alcohol. And if that binds, then you have the phosphate with -1, and you have the acid with another -1, and the amino group with a +1. That adds up to be a net charge of -1. So just realize that the net charge includes the -1 from the phosphate in all these cases. You can actually have a glycerol, another glycerol on the other side of the phosphate, and then you can have some weirder things, like you can have this phosphatidylinositol 4,5-bisphosphate. This is a sugar you did not have to memorize, but you might run across it in cell biology. And it turns out that it's a sugar with 2 phosphates on it. Each of those phosphates has a -2 charge. So this ends up being a net charge of -4. That's more complicated than I want you to necessarily memorize or remember. The other things are much simpler. On the bottom you have cardiolipin, which is actually a phosphate group that is connected to a whole nother 2 lipids. You can have it connected to a glycerol, which is connected to a phosphate, which is connected to another glycerol, and it's connected to 2 more chains. That's our version of a double-headed lipid. It's not quite the same as the archaeal version. Why in the world do we need a complicated double-headed lipid like that? Well, we'll get around to it at the very end of this chapter. It turns out that it's important for mitochondria. So you can forget about those for now. Just remember there are some complex structures, but you really only need to know and study the less complex structures that you can actually memorize and draw. So remember that we've had the DPPC, and the C in that is choline, and there we have the choline on the phosphate on the 2 tails, and that is correctly charged. Notice how the overall charge of this molecule is neutral. Net charge is 0. So sometimes remember that we showed you sphingosine before, and we said it was sort of a built-in chain. So you see sphingosine sort of combines a 3-carbon glycerol-like backbone, and then it goes right into a chain. There's no linkage at all here, not even an ether linkage, much less an ester linkage. It's very hard to break this off, almost impossible, because it's covalent. The other thing about sphingosines is instead of an alcohol here, they have a nitrogen, which forms an amide linkage to the fatty acid, but it works basically the same as an ester. So you can have the fatty acid going on and off. For all the ways in which this looks different, I want you to see how it looks the same. It basically has 2 tails going down, 1 head group going up. So as far as the overall shape of the molecule goes, this has the same shape as a regular membrane lipid. And so that the sphingosine is a built-in chain, and just notice that it has an amide linkage rather than an ester, but it works pretty much the same way. There's the hidden glycerol if you want to look for where is the structure that is most like the glycerol backbone. There it is. So sphingolipids have their own head groups. In fact, if you just had the sphingolipids, oh, by the way, you have this head group substituent, and you see the blue X right there. What can the X be? And the X can actually be just an H, in which case you call it ceramide. This is an important sphingolipid. And then there's sphingomyelin, which you might have heard of in other classes. This is our old friend phosphocholine. So if you have a sphingolipid with a phosphocholine, that has a special name called sphingomyelin. It's actually a very important lipid in the body and in some diseases. The other thing that you can see is that some sphingolipids, for their X, they have complex oligosaccharides. And so instead of a phosphate head group, what they'll have is they'll have a bunch of sugars. Still going to be hydrophilic, still going to be pointing up. And in fact, you can see some of the shapes that you've seen before. And there's one sugar, that's the purple diamond. And I told you not to memorize sialic acid because I can't memorize that. I don't want you to memorize that. But if you look at this, you'll see that it's important to the cell because you have the sialic acid on the oligosaccharide they've shown. Commonly, sialic acid will be on the outside of cells as the head groups of lipids that are in the membrane, pointing out. So when you think about these, just remember phosphatidylcholine, which is a glycerol backbone, and sphingomyelin, which is a sphingosine backbone, they pretty much look the same. In fact, their head groups look identical. From the outside of the cell, they look exactly the same. And on the inside of the cell, what really matters is how many double bonds they have, and you can switch those in and out. So the shape of what they are, when you look at these two things, they look pretty similar from a distance. So they function the same way in a membrane. But it's really important that they, because the glycosphingolipids have these different sugars on the front, they can have different sugars depending on the blood type. And this is what determines blood type. Glycosphingolipid glycosylation is the distinguishing thing between O, A, and B blood. So in fact, there are 3 different antigens. And because you have 2 copies, you can have one or both of these, or maybe none. Because it turns out the differences between them are very small. The O antigen has this oligosaccharide on it. The A antigen has that oligosaccharide, which is the same thing as that, but it has a GalNAc on it. And then the B antigen has a gal. Instead of, it's just not a NAC. You know, it doesn't have the NAC. If you remember your sugar structures, and this is where I say that it's important to review them because it's important for chapter 10 and 11 for you to even have this in your mind. But you should be able to remember galactose is a pretty common sugar, and the only difference between A and B blood is there's a bunch of N-acetyl groups on the A blood, and there's a bunch of, not just plain galactose on the B blood. There's so many of these, and blood is so prevalent, and when you give a blood transfusion to someone, antibodies will form against these if you've never seen them before. That's why you can't give somebody who's used to the gal a GalNAc blood. If somebody's used to the B antigen only, if you give them the A antigen, their immune system will freak out. The interesting thing is that the O antigen, if you don't have galactose at all, then the immune system won't freak out. Basically, you can give the O antigen blood to somebody who has A or B, and their immune system won't freak out because there's no galactose to freak out at. Exactly why that works is a little bit of a mystery to me, but I think it's pretty intuitive that the O antigen doesn't have anything that will make the others freak out, whereas if you give A antigen or B antigen blood to an O antigen person, they will freak out. So O is a universal donor, and AB blood is a universal acceptor. Okay. By the way, if you think about positive or negative, that's actually another molecule on the surface of red blood cells, and it has to do with, I believe it's a protein, and it's just, that's more of an immunology thing. But at least we can explain with biochemistry. The biochemistry of A versus B versus O is actually this simple. And so, for example, you can have immune receptors that interact with blood cells, and what they do is they bind these sugars. For example, there's a receptor called DC sign, and it recognizes the A and B blood antigens. And what it does, you can see the 1, 2, 3, 4 sugars that they have, the same 4 sugars. The fucose is also red here. And here's the GalNAc, so this is showing A antigen blood. But what it does is it uses this orange valine, sort of sticks between the sugars here. And the NAC part, the N-acetyl group, is sticking up into the solution. So DC sign doesn't care if you're A or B blood, but it probably will care if you're O blood, because it does interact with this yellow sugar, and people with O blood do not have that yellow sugar. Okay. So the question is really, if it really is this simple, the difference between A and B and O, can't we fix that? Can't we have chemistry that goes in and changes this simple chemistry? And the answer is, well, we might finally be getting to the point where we can actually do this. As it stands right now, we just need to collect blood from these different people, and we can only give the right kind of blood to the right kind of person. But the type O is the most in-demand blood because you can give it to anyone. So it'd be great if we could reliably convert A blood or B blood into O blood, which would just be a question of removing that end glycosylation. And so, by the way, what we showed you in the textbook, it's a little more complicated than that in real life, and you'll see some of those complications in these pictures. But mostly I'm going to give you this handout, and I want you to see how converting A and B to the universal donor will be clipping off the end so that it looks like this. Look at this, the O antigen. You have the fucose, and then you have the yellow sugars, and then the blue one. It's the same pattern here. Fucose, yellow sugars, blue one. This is simplified a little bit, but it has the same idea that you're clipping off those ends of it. The complexity is that actually some of the antigens grow beyond what this figure shows. You can see that there's a few extra antigens on. But essentially, if you can cleave them down to the O antigen is what does work for everyone. So it's a matter of cleaving off the extra stuff. Okay. So what they did to find this is they did not do protein design. What they did is they did sort of bioprospecting. They looked at a particular bacterium that they knew degrades mucin, which is an oligosaccharide containing molecule. And they knew that the bacterium degraded this, so they found the enzyme. They're looking for the enzyme that degrades this mucin, which has sugar molecules that are almost exactly the same as the type A and B red blood cells. So they looked carefully, and they were able to find an enzyme, or a couple of enzymes, that were actually able to bind to and break the exact sugar bonds that they wanted to break that would convert A and B to O blood, that would break off those edges. So this is a bit more of a zoomed in picture. This shows you them looking for the human gut bacterium that will do this, and they purified out A converting enzymes that would bind and break A antigens, and B converting enzymes that would do the same for B antigens. And the idea is once you break off enough stuff, you end up with the O antigen. This is the figure that I will print out and give to you in class. This might be on the test. I want you to look at this carefully because you probably can't see it right now, but I'll give it to you on paper because this is really cool. It shows you the sugar structures in the same way that we've talked about them, and it says exactly what the enzyme does and exactly what the real life antigens are. A little more complicated than the textbook shows, but similar enough that you can understand the strategy from what we've talked about. So look at those. Those are alpha and beta linkages. Those are the different kinds of saccharides that we've talked about. This will help you review and bring things together for the final. The other side of things is the enzymes. They have the structures of the enzymes, so they can see how the enzyme binds the sugars and where it cleaves them, where the catalytic machinery is. And so this is amino acid review and interactions review. So this is a great review. I'm going to print out just this for you. It's not a whole paper to read, but it's a highly technical, highly structural figure. But if you can take this apart and understand what's going on with this, you can see, oh, there is a histidine. Oh, that sugar is the galactose or whatever. That's what I want you to be able to do. So I got this from a poster online who talks about the reason why the need for this became even more apparent during COVID, because there were severe blood shortages. And he has a little history of people trying to convert blood types. There's a 1982 study that used just a regular old galactosidase that would convert B-type cells into O-type cells. And it actually worked. If you applied this enzyme and cut those sugar bonds, it would actually make O-type blood that could be given to anyone, and people with all of them. And people survived. It was really inefficient. The enzyme did not work very well. Lots of enzyme was required, and there's no way that the cost effectiveness worked out at the time. But it showed if we could get a better enzyme, maybe this could work on a higher scale. 2007, Nature Biotechnology paper had improved glycosylase enzymes, and they actually redesigned them, and they found some that would actually remove A and B antigens, neutral pH, low consumption. These seem to be hardier. Still not good enough. But in 2019, instead of designing and doing biochemistry by tinkering ourselves, they let evolution do that work. And that's when they screened the bacteria in the feces of an AB blood type person, and they found even better enzymes. And that opened the door to this most recent study where the enzymes look like they might actually work. So just notice right here, and by the way, look up here, the O-type antigen is sometimes called the H antigen. The type A antigen has, and this shows you the same thing, you have N-acetylglucosamine is the square, that's on type A. Galactose, just plain galactose, is B antigen, and you see all the linkages, and this is all the sugar stuff we've talked about. So on the test, I just want you to bring all this together. It's a good way to bring together multiple things that we've talked about. Be able to look at a figure like this, be able to maybe match it to a structure if I give you the structures, and just be able to match the figure to what's really going on. Notice why the enzymes have different names, things like that. So it's not just those enzymes that's what we can use for enzymes that modify blood types. There's lots of enzymes. In fact, snakes and venom, I think I had this. There's phospholipases in venoms that will break apart the esters that you see. And you see here there's a phosphoester breaking one, there's regular ester breaking ones. The differences between them are not that important. Just know that there are actually, and it's not just venom, there's real enzymes in your body, native enzymes, that you use to actually break down lipids to send signals. That's because phospholipids are important for signaling. This particular phospholipid, you see it has the inositol sugar on it. Like I said, that's important in cell biology. And to send or stop that signal, you make a lipase that just hydrolyzes the phosphoester, or in some cases hydrolyzes the regular ester, that will break down that lipid and stop sending the signal. But it turns out if it's good for you, that it can be used for ill by someone else. And there are venoms that have phospholipases. These are such effective and important signals. Some of them are important to neurons, and some phospholipases have involved in like snakes and things like that will actually go and break down the signals when you don't want them to. And it's very effective. That shows you how important these signals are. If you have a lipase that breaks them down, then sometimes you can have severe nerve damage or even death that results. And it's just a lipase. So that signal is important. If it goes away, that's not good. All right, one more, and then we are ready for the weekend.